Hi guys, welcome to another edition of Ringler Science. Hey! Coming to you from my basement of science. Today we're talking about deoxyribonucleic acid, also known as DNA. A student of mine recently asked me,
Mr. Ringler, how come I look like my mother, my sister looks like my father, and the two of us look a lot alike?
the simplest answer is, it's in your DNA.
Ah.
DNA is a tiny instruction manual, and there's a copy of it in the nucleus of every cell in your body, and it tells your cells how to grow. Today, we're going to make a model of DNA. To make the model, we're going to need some materials. We're going to need a cardboard tube, a manila folder. We'll need markers. I got a whole bunch of colors to choose from. I need a ruler, something sparkly preferably, and then some Scotch tape. To start out, I'm going to take a black Sharpie and I'm going to find the seam of the cardboard tube. I'm just going to copy the seam. Next, I'm going to take my scissors and I'm going to cut right along that seam. There, so now we have this. Great. Now I'm going to flatten this out and I'm going to take my ruler and I'm going to draw a line right down the middle of this so I can make two sides. And I'm going to cut it right down the middle, down that line. There. So now I've got the outer framework. All right, next I'm going to lay down these two right next to each other. I'll try to line them up. I'm measuring out every 2 centimeters. I'm putting a line and then I'm going to draw a straight line across so I know where to attach my base pairs. Marked all the rungs 2 centimeters apart all the way down to the end. Now I'm going to make my base pairs. Measure this. I've got 10 centimeters here. I marked it 5 in the middle. And now I'm going to go 20 centimeters tall. And if I did it right, it should be 10 wide again. Now I'm going to mark in 1 centimeter from the end. Okay, so these will be the tabs upon which we will tape these onto the spine, and then the base pairs will be here. We'll mark every 2 centimeters. And for the base pairs, we have adenine, A, pairs up with thymine. And that could go either way. It could be AT or TA. All right. And then we have cytosine with guanine. And of course, you can flip that. It could be guanine with cytosine. Right, and we'll go all the way down. A and T always go together. T and A always go together. C and G, or G and C. Now we're going to color code it. Adenine will be red. And I'll make all the adenine red. And then we'll make all the thymine blue. We'll make all the cytosine green. And I'll make all the guanine. Let's go yellow. So I've got that and it looks pretty good. I'm going to cut that out now. And then I'm going to fill all this in on the other side so it looks good when I build the model. All right. All right, so I made a second sheet of these. I made about 24 base pairs. Now we're gonna cut these apart. All right, I did them on both sides, you'll see. And we'll cut them up now. All right, we're on to the last step, where we attach the base pairs to the sugar and phosphate spine. All right, so I'm gonna kind of put them on a diagonal right there, line it up, glue it down. All right, so it does take a little bit of finesse to put this thing together. So I'm using glue and Scotch tape. I've got my base pairs on one side and I'm putting it onto the other side now. It's pretty tricky. Oh yeah. This DNA is coming together. So, here you go. Here's my model of a strand of DNA. Pretty good. We've got the double helix, the twisted ladder, we've got the colorful rungs. It's our DNA model. And that's all. Well, thanks for coming to another edition of Ringler Science.
Hey!
I'll see you next time.